So this is part 4 for physiology, where we're going to deal with propagated action potentials. So we're just continuing our story that we started a few times ago. So in particular, we're going to look at how we move action potentials from the dendrite towards the synapse. So the story so far, we have this thing called a membrane pump. And what it does is it moves ions from one side to another, or any other molecule. And in particular, we're going to deal with the sodium-potassium pump. And it moves sodium ions outside, moves potassium ions inside of the cell. When it does that, it sets up concentration gradients. Every positive comes with a negative, so when potassium ions get the opportunity to leak out using a potassium leak channel, they leave behind their negatively charged partners. That makes the inside of the cell relatively negative compared to the outside. Because there's an attraction between positives and negatives, we call it Coulomb's law. The potassium ions eventually stop leaking. We call that the resting membrane potential. What does it look like? Something like this. Or excuse me, what we do is we look at, well, what's the big point? Why do we care about all this? Again, it's because of reflexes. The big deal with all of this is reflexes. And when we look at reflexes, we have to deal with 5 components. Those first of those components is going to be what tells you, by the way, you touch something hot. We call it a sensory neuron or a sense receptor. It's going to send that signal down along the neuron to a processor. We call it an interneuron, which will then say, hey, motor neuron, or the doer, do something, and then we'll get a muscle to contract and you pull your arm away. When we look at that, we can actually end up making a graph. That entire process there is referred to as an action potential. So action potentials have 2 components. We happen to have a depolarization and a repolarization. You happen to have something that triggers some type of increase in sodium ions inside of the cell. What that will do is make the inside of the cell less negative. When we make the cell less negative, eventually it will reach a potential called threshold. Threshold is when we're going to have a sodium voltage gate open up. When a sodium voltage gate, it opens because of the voltage or the membrane potential. When that opens up, we'll get a depolarization and we'll get a rapid influx of sodium ions from the outside of the cell into the cell, making the inside go from negative, relatively speaking, to positive. When you have this positive membrane potential, that's going to cause the sodium voltage gates to close and open up potassium voltage gates. When the potassium voltage gates open up, that's going to allow for the efflux, or the leaving of potassium ions from inside of the cell out. When that happens, we're removing positive charges from a positively charged area, which will result in a negative charge again. The catch is the potassium voltage gates will close at a lower potential than our resting membrane potential, thus causing this little state down here at the bottom that I'm circling with the mouse that we call the refractory period, or a state of being hyperpolarized. What that will allow for is action potentials to move in one direction and not 2. Thus, there's a start and there's an end. This entire process here is what we call an action potential. There is no other version of an action potential. It's a depolarization, repolarization. There's no bigger version. There's no smaller version. It's just what it is. These turn out to move, and as we pointed out yesterday, a depolarization will trigger another depolarization. So when we happen to have the influx of sodium ions, as you can see with the mouse maybe a bit right here, when we add those positively charged ions inside of the cell, that makes the area next to it positively charged, or at least more positively charged, less negatively charged, which will trigger threshold, open up the sodium voltage gate, sodium ions influx, thus changing the area right next to it to becoming a little less negative, a little more positive, until we reach threshold and we open up that sodium voltage gate, and so on and so on and so on. What we end up getting is a series of depolarizations, or a wave of depolarizations, that we either call a moving action potential, or a propagating action potential, or a nerve impulse, or a wave of depolarization. There's all sorts of terms that we can use to describe one depolarization triggering a whole bunch of others. So if we jump into the brain, we have all these neurons. These aren't accurate because they're not convoluted enough. But these light signals that you see moving along, that would be an artistic way of saying action potentials. So what we get is sodium ions outside, they go in, we shove them back out, we just get this random fluctuation. Eventually what they're going to end up doing is reaching the ends, and that's actually where we're going to end up causing this action potential to connect to another cell, but that we don't need to deal with just yet. So when we look at the structure of a neuron, it turns out to have some rather basic parts. So we happen to have where the nucleus is, we call that the cell body. There's going to be a whole bunch of attachments to the cell body, we call those dendrites. Dendrites are where, for the most part, we're going to receive information. So information goes into dendrites, gets processed at the cell body, collects right here at something called the axonal hillock, even though it's not labeled, and then it's going to move along a very long process that we call an axon, eventually going to the axonal terminals or the terminal buttons or a whole bunch of other words that go there, where we'll end up moving that information somewhere else. The flow of information will be from dendrite to axon terminals every single time. Not all the neurons turn out to be built the same way, and not all axons will look alike. So what we end up getting is a whole bunch of different variations on neurons, and the reason for that is they do different functions. They will behave differently. One of the things that we notice when we look at neurons is they happen to have layers around them, and we call that layering myelin. Myelin is just a whole bunch of fatty tissue, it's cell membranes that come from one of two different sources. If we look at something that we call the peripheral nervous system, which is a nice way of saying your nerves, the source of all that myelin will be a type of cell called Schwann cells. If we look in your central nervous system, which is a nice way of saying your brain and spinal cord, that myelin is going to come from a type of cell called an oligodendrocyte, which just means a many-branched cell. Both of them will do the same thing, it's just different ways of getting to that answer. Why do we care about this myelin stuff? Because it turns out myelin allows for the propagation, or a more rapid propagation, of an action potential. So what we would normally have is we'd have a depolarization, then 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 a depolarization, and that would move along at a certain speed. The catch is, because of the way that charges work, I could actually look and see, well, how far out does that depolarization affect another depolarization? How far does its influence turn out to reach? And it turns out the length of a piece of myelin, even though myelin's misspelled, the length of a piece of myelin perfectly matches that distance. So we get these gaps between the myelin, we call them nodes, and what we can get is if we add myelin, we will make it so that a depolarization will influence another depolarization one node away, which will influence another depolarization another node away, which will have another influence another node away. And the result is it looks like the action potential jumps around. The result of it looking like it's jumping around is what we call saltatory conduction, because salta means to jump or to dance. Why do we care about adding that? Because it rapidly increases the rate of action potential conduction, which means if we're having your hand touch something hot, the figuring out that something is hot turns out to be far lengthier than it is to just to say move your hand away. Because to sit there, put your hand on something hot, say, oh, that's hot, I should move my hand away, move your hand away, takes too long. If we can send a bit of information that says yank your hand away, then figure out that it's hot, this is potentially life-saving. And that's what myelin allows to have happen.